Everybody, it's Miss Peachy from WCA Biology, and today I'm doing a little recording here to help you out with your portfolio on Wednesday, which is the effect of cell size on material. First thing, when you have any portfolio, so you can see here on your planner, the one place I think would be a great place for you guys to make sure that you go to access materials for that portfolio is my website. Remember to access my website, just go to that push pin icon at the top of the screen. And when you do that, you will see some section message boards here. Biology is one of them. We'll click on the bio one and you'll just follow the links here where it says link to Mrs. Peachy's website. Okay. Once you do that, that's going to take you to our website here. It'll open up on the home screen, but you're going to click on Biology A at the top of the screen to access this page. When you scroll down, you'll see portfolio instructional videos, and that's what you need to be looking at. So we'll have a video here for the cell size portfolio. This is an older video that I have up here, which I'm remaking at this time. And then this is going to be the worksheet that you need to do. Now, several of you guys struggled last portfolio because your PDFs weren't saving properly. Using this pop out here, this download is going to prevent you from having that issue. Okay, so go ahead and take advantage of this. When you open this up, you will be able to download it to your computer as a Word document. And if you can't use Word on your computer, say you don't have that program, you can just go File and make a copy. And when you do that, then you'll have the ability to access and edit the document in Google Docs, which is what I've done here. Okay. So you can type in this, you can make modifications to it. And then when it comes time to finishing it, then you would go to File and Download, and then download it as a Microsoft Word file to your computer. Even if you don't have Word, you can save it on your computer, and then you can put it in the Dropbox from there. That way, even if you don't have Word, you have the ability to still save it as a Word document and get it to me as a Word document so that I can then do what I need to do to view it. Okay? So that kind of is the technical parts of how to get at this document. I would highly advise against trying to modify the PDF and save the PDF because it comes with all sorts of problems. Okay, so basically what you're doing here is you're looking at how the size of cells is going to kind of be limited naturally. Like, can a cell be too big? And if it's too big or too small, is that going to limit the function of the cell? The idea really is that a cell is kind of like a cube, and each side of the cube is how it's able to access the outside of the cell. It takes in nutrients through the sides, and it gets rid of waste products through the sides. But it has to feed the entire volume of the cell. So a cell like this is a very big volume compared to a cell like this, right? It's a lot bigger of a volume. So what we're looking at today is something called surface area to volume ratio. And we'll be kind of going through those calculations a little bit later. So for this, you need to have a potato, at least one large one or two medium-sized potatoes. I was able to use just one large potato and it worked fine. And you have to cut that potato into squares. And you need a metric ruler here because you're going to measure your largest square is going to be 3 centimeters by 3 centimeters by 3 centimeters. And that's what I've done here. And then you need to make one that's 2 centimeters by 2 centimeters by 2 centimeters, one that's a 1 by 1 by 1, and one that is half by half by half. So 4 different potato cells. The other thing I would really suggest is that you soak these potatoes for a while in warm water to kind of get the starch out of the potato. It helps to make the potato more porous, and it helps to kind of make this work a little bit better. Once you've soaked them, put them on some paper toweling or some regular towels and just kind of dry them off so that so they're not like super wet or anything, right? We want to make sure they're fairly dry. The other thing you're going to need is you're going to need some food coloring. And I'm putting it into just one jar. This is like a regular cup from my cupboard. And I put quite a bit of blue food coloring in this, and then I filled it with some warm water. Warm works a little bit better than colder water. I also have a pair of tongs here because I don't want to get my fingers all blue. And once you have all that, then a timer essentially, and we'll kind of see what happens next here. So I'm going to take my video camera and I'm going to put it down on my desktop so you can kind of see what I'm doing. Just a second, my mouse isn't being very... Okay, so now you can kind of see from the vantage point if you were doing this experiment yourself. So we are going to take well, first of all, I also put some lemon juice inside of the container. It does say to do that. It's kind of like dyeing Easter eggs. The acid in the lemon juice is going to help to kind of open the pores up a little bit there. So what we're going to do is we're going to take those cells. This is my large one, and we're going to put them inside of the cup of food coloring. Okay. I can put all of them in a single cup. Just kind of arrange them in there a little bit so that they can all fit. They should all be sunk. There we go. The little guys are a little easier to get in there. Okay. And basically we're going to wait. I believe if I look at your directions in here, I think it says to wait like 10 minutes or something like that. Let me just double check on that. 15 minutes. You might find 15 minutes isn't like adequate enough. Sometimes it does work to let it wait a bit longer. So I'm going to set my timer for 30 minutes. And I'm obviously going to pause the video because I have no reason why you're going to sit here and just watch a couple of potatoes in a jar for 30 minutes. So, but I'm going to set my timer here for 30 minutes. As you can see here. And we'll come back and check on this in 30 minutes. All right, it has been 30 minutes, so let's kind of see what we have here, shall we? We'll go ahead and take out our cells out of the water, and we'll set them on our paper towel here to just kind of pat off a little bit. It's a little fishing game, huh? The little guy is going to be kind of a pain to get out, I believe. There we go. All right, I got to grab a knife real quick here. All right, so they are now on paper towel. I'm just kind of patting them dry a little bit here so that they're not too messy. So the idea here is that we're going to cut these guys and we're going to see how far they penetrated into the cube. Okay. So let me go ahead and cut the first one. Okay. And I mean, you can see that it didn't penetrate super far, but if we look at, it's real dark on the edge, it kind of goes about that far in. I'll zoom in on that in just a second. We can look at that. Okay. Look at number 2. Okay. Our second one. our third smallest. And then our fourth smallest. This little baby guy here. Okay. So it really doesn't penetrate very far into any of them, and that's okay. That's really not the point. So we'll kind of figure out what the point is here in a moment. Let me try to zoom in on these for you. See what we can do here. Let's get the big guy underneath the camera so you can kind of get a better view. Okay, so it's not super focused, but let's try to see if we can focus it a little bit better. There's some fancy good camera here. All right, the idea is here, it's going to be millimeters we're going to be looking at, right? So if I look at how much blue is in here, I can kind of say it's going as far as like right there-ish, right? It's a very light hue of blue. So it looks to me like I'll go this direction so y'all can see it better. But it looks to me like if I put the 0 on the edge, that maybe we're talking about a millimeter or potentially 2. Okay, there's 0. And then I would say maybe 2 millimeters in. Wish my camera wasn't quite so fuzzy, but try that again. There. It's better focused that way. There. But I mean, it's difficult to see. But 0's on the very, very edge. So we're going in maybe, let's see if I can do it right here. There. 2 millimeters. Did you guys see that at the top there? I see it. 2 millimeters. Okay. So we're going to type that in here. So this is my 4 centimeter one. Just going to move that out of the way. We're going to go here. It's 2 millimeters, which is a millimeter is a thousandth of a meter. So 0.0. I'm just going to put 2 here. Millimeters. I don't think it really matters if we do this in the same units as the other stuff, just because it's really not something we're using in a calculation. It'll just be easier that way. Okay. So let's do the same thing. Okay. This is our second biggest one. I fully expect this to be about the same, by the way, ladies and gentlemen. So I think it's about the same, don't you? Like if I see where the little blue shadowy part, which I think you can see better up on top here, kind of ends, I would say it's around 2 millimeters as well. I mean, maybe a hair more, maybe 3. So we can go with 3. But I don't think it's going to be a whole bunch difference. Right. We'll figure out in a minute. Okay. Here's our second smallest one. And again, roughly 2. I see that, roughly 2. So I'm just going to go back and do 2 millimeters on that guy too. Goodness, sorry about the little coughing thing. And a little baby guy right there. Get him in the view. You can see him. He's so tiny. It wasn't as perfectly straight as the other guys, though. That one's a little bit harder to see because you're so tiny. This side is probably a little bit better. I mean, it's roughly the same as well. About the 2 millimeter mark. Oh, we can't even see the guys off the camera. Sorry. I mean, this is he's so small, it's really difficult to see. Let me scooch it. So I'd say it's roughly, keep sticking to the edge, but it's about the same. About the 2 millimeter mark too. So we're just going to put that down too. I mean, honestly, they're all about the same. And that is totally expected. Okay. But what I want you to notice this is what I really want you to notice. Okay. Instead of looking at how far the food coloring traveled through, I want you to denote how much is still white. Like, look at how much of this cell is white. All of the cell is starving right now because no food got to that part of the cell. When you get to your medium-sized, kind of your size 2 here, again, quite a bit of the cell is still starving because the food didn't penetrate down deep enough. Less for this guy. And when you get to your little baby, I mean, just a very small part in the middle didn't get blue. Otherwise, I mean, it's the edges, yeah, for sure. And if I would have left that so longer, hopefully it would have penetrated more. But the idea here is that there's just a small part in the middle compared to my big one. This guy's like the majority of the cell's dying. This one, you're getting to a point where you're getting more saturation. If you think of the blue food coloring as the food trying to get into the cell, you can really think of it as, how much of that cell is getting fed. And it's a lot less for the big one. So let's do a little bit of math, shall we? Okay. So our first cell 1, has a length of 4 centimeters on one side. Surface area. Surface area is the area of all the surfaces together. So a cube has 6 sides, right? Each side has a length and a width. So we did, what was our big guy? Oh, I'm sorry, he wasn't 4, he was 3. My bad. Our big guy was 3. So he's 3 by 3. 3 times 3 is 9 square centimeters. But there's 6 sides. So we have to multiply that times 6. So the surface area of our big guy was 54 square centimeters. So the surface area here, you're going to take, I'm going to put the little formula in here, length times width times 6. Oh, length times width times 6. That's how you're going to get your surface area. Length times width times 6. Okay? So do that for your other ones. For cell number 2, 3, and 4. Remember, cell number 2 had a surface area of 2, 3 was 1, and this guy was 0.5. Okay? Let's do volume. Volume is length times width times height. So for this one, 3 times 3 times 3, because it's a cube. 9 times 3 is 27. This is cubic centimeters. Okay? And then there's the hard part here, surface area to volume ratio. We take the 57 to 27 is our surface area to volume. We then just need to reduce that. I'm sorry, 54 to 27. We need to reduce that to lowest terms. 9 goes into 54. 9 times 6, right, is 54. 9 goes into 27 3 times. 3 goes into 6 2 times. 3 goes into 3 1 time. So the final reduction here is a 2-to-1 ratio. Okay, so you just have to reduce that number. This is our surface area number. This is our volume number. Just reduce it down to lowest terms. And we put it as our ratio of 2-to-1. Okay, so now I'm going to leave you to calculate the surface area and the volume for cubes 2, 3, and 4, and then the surface area to volume ratio. Now, they pretty much all traveled the same distance with the food coloring. But remember, it's not necessarily about the distance traveled so much as how much of the cell was left empty. So when you think about it, the smaller the cell, the more of the cell received food. The larger the cell, the less of the cell received food. So think about that when you do your analyze and conclude questions, okay? So now you need to complete the pre-lab work. Make sure you answer those questions, finish the data table, and then do your analyze and conclude section as well. Okay, so those are the sections you need to do. And I'm going to leave you there. If you have any further questions, just let me know. Otherwise, I hope you guys have a great day.